Hello everyone. In this video, I'm going to show you one way in which you can determine the knockout efficiency from a Cas9 mutation experiment using nanopore sequencing and tools that are built into Galaxy. Now, before I get started, there's several caveats to mention here. First of all, nanopore sequencing is error prone, so we should not expect these results to be perfect or even that highly quantifiable. Okay, but it should give us overall a rough idea of how many of our target sites have been modified and how. Um, Also, many of the tools we're going to use today in Galaxy were made for reads that come from Illumina or PacBio sequencing, not necessarily from Nanopore sequencing. So there may be some suboptimal things that are happening in these algorithms. We're still going to get a result, but I need to tell you all of this just so you know that it is not it's not exactly perfect what we're about to do. Okay, but it is something that can give you a rough idea of how well you did with your Cas9 experiment. Okay, so with all that out of the way, here's the type of experiment we're going to analyze today, in which we have a target gene, and we're going to cut it at three different locations using Cas9. So there's going to be the same Cas9 throughout, but then three different gRNAs to cut here at sites 1, 2, and 3. The potential products of that reaction are going to be numerous, right? We could have large deletions between sites 1 and 3, 1 and 2, or 2 and 3. We could have small insertion deletion mutations at any three of those locations as well. So all of these things we need to keep in mind as possible products because they're all going to affect the expression or translation of our gene in different ways. Okay, so we want to be able to capture all of these different possibilities. Now, the way we're going to do that experimentally is by designing a forward and reverse primer that bind outside of these target regions, okay, such that we can take these cells that have been treated with Cas9, extract their genomic DNA, and use PCR to amplify any type of target sequence that exists between this forward and reverse primer. So you see what we've done here is located the forward primer upstream of the first site for Cas9 and the reverse primer downstream of the last site. Okay, so that means that even the wild type and mutants, so whether they be indels or deletions, should all be amplified via PCR because the Cas9 won't really affect the binding of these primers. Okay, so with that in mind, we can run this PCR and we're going to get somewhat of a distribution here. We're going to get some of these amplicons that are pretty short compared to the wild type because of those large deletions. And we're going to take that mixture of amplicons and submit it for nanopore sequencing. So we choose Nanopore here because most of the commercial services for amplicon sequencing use Illumina, and they require multiple days or a week to return data, and they're relatively pricey. So I think GeneWiz's AmpliconEZ service is around $80 now per sample. Whereas nanopore sequencing, we can get those results back next day. You can do this in real time in your own lab if you have one of these sequencers as well. And the price is right at about $30 per sample. All right. So we're accepting a little bit of error here to get the data back faster so that we can utilize these cells earlier on after they've been modified. Okay, but with that in mind, when you use nanopore sequencing to analyze these amplicons, there's going to be some garbage that's included in those reads. So you're going to get some reads that are just way too short. They're truncated, so they don't even include any of these target sites. So they shouldn't be included in the analysis because they're largely inconclusive. You don't know if the rest of that sequence would have been mutated or not. You may also see concatemers here. This is mostly a byproduct of PCR, actually, where multiple bands are joined together. And so in those reads, they'll be, they'll be longer than expected and contain multiple copies of these target sites. So we'll need to clean up the data to get rid of these reads that are too short, too long. Also, there's going to be some reads that are just low quality. That's the dashed line down here, where the sequencer wasn't highly confident in those bases to begin with. So we want to discard those reads because that might make us think we have mutations where we actually do not. Okay, so like I said, we're going to have to clean up the reads, but we need to establish some bounds for that. So obviously, an acceptable read would be something that's the whole length from the forward to the reverse primer, but it really shouldn't be longer than that. So we need to know the length between these primers for anytime we're doing this analysis. Okay. Also, we would accept reads that are as short as a length that would be the whole wild type read, but accommodating the deletion. So if this is a 500 base pair read, for example, but my Cas9 target sites here, if they're all chopped up, then that will remove 150 bases. Then my minimum read would be 350 bases long, okay? And maybe we shorten that a little bit to allow for additional bases being deleted at each cut site. Okay, so we're going to accept reads between the minimum expected length and the maximum expected length. We're going to get rid of low quality reads. We're going to get rid of truncated reads, incomplete reads that don't have the entire sequence. What I mean by that would be a little bit of sequence around the forward primer, a little bit of sequence around the reverse primer. Okay, so we're going to filter those reads. That is step one here in our strategy. After we do that, it's important to note that the reads you get back from Nanopore are going to be in random orientations. So some of them are going to read the coding strand, some of them are going to read the opposite strand. So we need to align all of our reads in Galaxy to our expected wild type sequence. So we're going to do that in step two. After we do that, then we're going to do another modification here. The motivation for step three is that eventually what I want to do with all of these reads is translate them from DNA into amino acid sequences, because that's what really matters in a Cas9 experiment, right? If I swap an A for a T in the DNA sequence, that might be a mutation, but that might also be a silent mutation that doesn't affect the amino acid sequence at all. So at some point here, I need to take these reads and translate them into amino acid sequences so I can see exactly how that protein sequence changes, and that'll let me know how many of these mutated genes actually have or correspond to mutated proteins. So with that in mind, here in step three, what we need to do is take all of these reads and cut them off such that they all begin with the first base of a codon and end with the last base of a codon, okay? So that way, when I take these reads and translate them in Galaxy, they'll all be starting at the right spot in the reading frame, okay? The reads that you get from Nanopore, they're going to start at random locations, okay? Especially if they're in that reverse orientation. So that's one thing we need to do here before in step four, we translate the reads into amino acid sequences. Then there's a BLAST-P that's built into Galaxy. We're going to use that to align all of our amino acid sequences to the wild type sequence. That's going to generate a tabular file that we can put into Excel and then analyze how many of these reads align perfectly or less than perfectly to the expected amino acid sequence. Okay, so that's the overview there of how we're going to do that. Now let's actually get started by looking at a specific example here. Okay, so here is one experiment where we're targeting this PML gene. Some things you need to know about PML. First of all, there's multiple splice variants of PML. And so you can see that here. So there's 1, 2, 3, 4, 5, 6, 7, maybe 8, up to 8 exons. And those are shown in the transcript sequence down here. So the first exon is in white, second exon in gray, and it alternates after that. You can see the start codon for the gene here, ATG. So that's the beginning of the coding sequence that then goes through exon 2. And in this specific experiment, we're going to be using guide RNAs from Synthago, or as now known, EditCo, that target exon 3. And so what I've done for you here is highlighted each of those guide sequences in yellow. Their PAMs are highlighted in lavender. Okay, so it's Cas9, so we have an NGG PAM, and our guide sequence is going to be the 20 bases on the 5' end of that PAM. Or if the PAM is on the opposite strand, so here we have a CCN PAM, it's 20 bases downstream of that. Okay, so as you can see, we're targeting 3 different locations. The cut should happen here, here, and here. Okay. So with that in mind, we're targeting this 3rd exon. I mentioned to you before, we're going to amplify this target sequence using PCR. I don't have my primer sequences shown here because this is a transcript sequence. So the intervening introns have been removed, but I can tell you our forward primer is a few dozen bases upstream of this junction here. And then our reverse primer, it starts right about there. Okay. That will give us an amplicon that looks something like this down here. So there's my forward and my reverse primers. They're highlighted in red. And what I want to emphasize here is that there's quite a bit of space between the forward primer and the first Cas9 target site. Same thing with the reverse primer. It's far removed from the last target site. We do this for several reasons. First of all, any sequencing technique is going to have a much lower quality next to the primers. So we want to make sure that those primers are far away from our target sites that we actually want to measure. Also, this specific product was designed to be used either for Nanopore or AmpliconEZ. So its maximum length there for AmpliconEZ is about 500 bases. Nanopore doesn't have that limitation, but these primers were designed with that flexibility in mind. Okay, so we're copying this entire sequence. So even if Cas9 deletes everything in this region here, our forward and reverse primers will still be able to bind, and we'll get a good read on this. Okay. Up here's our amino acid sequence. The base is highlighted in red. That's where our Cas9 target sites are. So those are the spots that we expect to see mutations. All right. And then coming back up here, one other thing I want to explain is I said as part of our analysis, we're going to take all of these reads, we're going to clean them up, we're going to align them, and then we're going to cut them off. So we're going to cut them to where they each start at the same reading frame. Okay. And so what I've done here is if you start here at the start codon and scroll down to this red text here, that red text is where we're going to cut off these reads. So everything before and including the red text will be removed from the reads, meaning that all of our reads should start with CAG. And if we check the character count here, I've included the start codon and everything up to that last red character. That's 732 bases, which is divisible by 3. So we know that this CAG right here should be an actual codon in the PML gene. So we'll start translating there and then move forward. Okay. So the red and everything before it will be removed. Likewise, on the other end here, the red target here and everything after it will be removed from each read. This isn't absolutely necessary, but I like to do it because that gives me a library of reads that are all exactly the same length. So that simplifies the analysis a little bit, as we'll see at the end. Um, but what I'm saying here is that every one of our reads, before we translate it, will be shortened to 243 bases. They will start with the codon CAG. So that should give us amino acid sequences that are 81 amino acids long. All right. And what our job is to determine if those 81 amino acids match the wild type sequence or if they vary significantly within the space of these target sites here. Okay. So some of them are going to be shorter, some of them are going to have mutations, etc. Okay. So without any further ado, let's jump right into how we're going to analyze that in Galaxy. So we go to usegalaxy.org, sign up for an account. We can log in and get a screen that looks something like this. Now I've prepared a workflow for this analysis here. And let me show you how that works. Okay, so in this workflow, the first thing we're going to do is input all of our files, which would be all of our sequences, and a few different parameters. Specifically, you're going to have your FASTQ file from the Nanopore sequencing. We want to upload that. We're also going to have a FASTA file that has the wild type DNA sequence for our gene. Okay, so these two things can be uploaded into Galaxy. I'll show you how to do that in a minute when we get started. But then these three fields here are parameters that we put in whenever we start the workflow. The first input here is the maximum read length that we want. So like we said before, the amplicon that we expect here is 500 bases. We're going to add a few bases to that just in case there was an insertion at one or more of these Cas9 cut sites. So you see there I have plus 2% of the wild type amplicon length. So let's take that 500 and make it 510, okay? We don't expect a lot of insertions, but let's include them anyways. And then we have the minimum length here, which would be that wild type length minus the distance between Cas9 target site 1 and 2, or sorry, 1 and 3. That would be the largest possible deletion, which is about 150 bases. That'll be 350 bases here then, but we'll subtract 10% from that, just in case there was additional deletions at either the first or the third Cas9 cut site. So we'll accommodate anything that's 315 bases or more. So that's our range here. We want to keep anything between 315 and 500 bases. Then we have a quality score that we need to input here. We're going to be pretty lax on quality. We're going to input a pretty low minimum score here of 20. If you wanted to be more stringent, you could increase that to 30, but I'll leave that up to you. Okay, so all of these inputs here will go into our first tool, which is this footlong tool in Galaxy. And what it's going to do is everything we just talked about. So in here, it's going to take that FASTQ file that we uploaded, delete anything that's longer than the maximum length, shorter than the minimum length, has a lower quality than what we input, okay? And it's going to use the wild type DNA sequence as a reference here. All right. After we clean that up, then we're going to align all of those reads to our reference, our wild type expected gene sequence, okay? Using this tool called Minimap 2. All right. Minimap 2 accepts an input of that cleaned up FASTQ file and the reference, and then aligns all those reads accordingly. It should be mentioned here that a lot of folks are probably used to using HiSat or bow tie for this type of alignment. But this is one of those cases where those tools were developed for Illumina sequencing, but here we have longer reads, and Minimap was designed with that, the longer reads in mind, okay? So that's why we're using this specific tool here. Now, the output of Minimap is going to be a BAM file. That's a binary alignment file. We can't really work with that, so we need to convert that into something else that we can understand. So we're going to put that through these tools over here, okay? Specifically, we're going to be using the SAM tools, FastX converter. What that does is it can input a BAM or SAM file and then convert it into a FASTA file. But at the same time, the other nice thing about this tool is that we can filter which type of reads we want to extract from that BAM file and put into a FASTA file. The reason we need to be aware of that is because the BAM file is agnostic to orientation. Well, I mean, what I should say is the output of this tool would be the reads in either the forward or the reverse orientation. You would get a mixture of those types of reads. But Minimap flags each read according to which strand of your expected sequence it aligns to. So Minimap does include in its output whether that strand is forward or reverse. There's a flag that's added to that each of those reads. And this tool here, the SAM Tools FastX, can recognize that flag. So you'll notice there's a fork in the road here. What I'm asking SAM Tools to do is create two FASTA files for me. This top one will isolate all of the reverse reads. The bottom one will isolate all the forward reads. Now, when I say forward reads, I mean it contains the same sequence, the coding sequence that I'm expecting. So it would go ATG ... right? So these are the reads. This is the orientation that I want down here with the forward orientation. The ones that are in the reverse orientation, we're going to take those and use another tool called reverse complement to get the reverse complement of all of those reverse reads, making them forward reads. So then we've got two FASTA files with forward orientation reads, and we're going to rejoin them with this tool, concatenate data sets. Okay, so you can see the inputs for that are the two different FASTA files. That gives me a single FASTA file output that I then modify further. So at this point, I've got all of my reads in the same orientation, okay? And this is the point where I want to cut them off on the 5' and the 3' end to ensure that they all start on the same base of the same codon in the appropriate reading frame. So for that, I use this tool here called CutAdapt. Now, this CutAdapt tool was originally designed to remove primer sequences and adapter sequences from Illumina reads. But we're going to use it here to identify a specific part of our amplicon. So for example, that text highlighted in red, we can input that into the program, and it will look for that in every read, and then delete everything, including that sequence, and on the 5' end of it. Okay, so we can use that tool to remove all of this sequence, and then we can use that same tool to put in a different sequence and say this is a 3' primer adapter, or whatever you want to call it, and we want to remove that and everything to the right, or on the 3' end of that, leaving us with only this sequence in between. Okay. Now, I should also mention that since this is nanopore sequencing, it's not perfect. We may look for this sequence and not find it in a specific read because maybe the machine made a mistake and instead of a C here, it recorded an A. So the other thing I'm going to ask CutAdapt to do is, if we scroll down here, I can have it filter out any reads that are untrimmed. So we click this from no to yes, and now if there's ever a read that doesn't contain that sequence, it will be removed. This should be a very small fraction of your total sequences, but it will simplify the analysis later on. We might be cutting out some valid sequences here, but the assumption is that that number will be so low, it's negligible, okay? So we take all of our reads, make sure they start at the same base on the 5' end. And by the way, here's an input there. That's for that red text on the 5' end. And then we take those modified sequences, do the same thing on the 3' end, and cut them off there. Oh, and by the way, I haven't mentioned this yet, but my strategy here for determining what that input sequence should be is I'm trying to place the first base about 50 bases upstream of our Cas9 cut site. So I want there to be plenty of information here that should be unaffected by the Cas9 mutation. All right, so I have this little constant region that should be in all of my reads, okay? And I can use that as a control, right? I can say if all the reads contain this sequence, then we know that they're valid reads, okay? If they didn't contain this sequence, then I might not, I can't use them for BLAST-P analysis. And you'll see how that manifests later. But this red sequence here is about 20 bases. That's 50 bases away from that first cut site. And it ends here at the beginning of one of the codons. Same thing for the red text here. That's about 50 bases away from that last cut site. Okay. And it ends, here, let's check our math. It makes these reads end, yeah, on a codon. 975 is divisible by 3, right? So that would end the read at residue 325. Okay. So that's what's going on in this block. We're taking all of our reads and trimming them down to the same exact length, starting at the same base. And by the way, for future reference, all of those reads should be 243 bases long, which means they would be 81 amino acids long as far as amino acids go, okay? And they should all have the same amino acid sequence up to about here, about, what is that? 17, the first 17 residues for most of these are going to be the same. But then after that is where we expect to start seeing these frame shift mutations where the sequences may vary significantly. We may see some reads that have the first 17 bases are missing all of these intervening residues and then resume having the same residues after the deletion. That's possible here because these cut sites are staggered by a distance that's divisible by 3. That's not going to be the case in all experiments, but it is here. Okay, let's get back to it. Once we have everything aligned and trimmed and in the same orientation, then we can run a blast analysis. So here, the first blast I have it doing is blasting the nucleotide reads against the wild type nucleotide or DNA sequence. All right, so that'll generate an alignment there. But what I really want is to translate those trimmed reads into amino acid sequences so I can do a BLAST-P against the amino acid sequence of my wild type protein here. This is the output that we're actually going to use to calculate editing efficiency. Okay, so that's the end of our workflow. All right, so that's everything from start to finish. And unfortunately, that's where it ends for us in Galaxy. There's not really any tools that help us work with amino acid sequences. Galaxy is built out for nucleotide sequences, but not so much amino acids. So we'll have to finish this workflow in Excel, but we'll cross that bridge when we get to it. Okay, so let's get out of here and show you how this would actually work. So first thing I'm going to do is put in a name for this history. So this is going to be PML AO analysis. There we go. Save that. All right. And then I'm going to upload my files. So in this case, I've downloaded the Nanopore sequencing data to my computer. So I'm going to click choose local file. And they are here. All right. So here's one of those files right there. This is the FASTQ data that comes from the Nanopore sequencer. Double click on that. The other thing that I need is going to be a FASTA file containing the wild type DNA sequence for this gene. And that is going to be here. So let me just open this up real quick, show you exactly what that looks like. So it's a FASTA file. You've got a greater than symbol, a name, and then a line break. And this would be the entire coding sequence of PML, starting with the start codon there, ATG, and continuing all the way to its stop codon. All right, so you can retrieve that from NCBI Entree, but that's the entire wild-type DNA sequence. I don't need to limit this to the target site or just what the amplicon covers. The entire gene sequence is fine to have here. Okay, so let's grab that. And then we're going to hit start here to upload those files into Galaxy. Depending on the time of day, the day of the week, this may take longer sometimes, but that's what we like to see is green bars. And then you'll see them pop up over here in your history as well, and we can proceed whenever they turn green over there. All right. But for now, I'm going to get my workflow started here. While those are getting ready, you can see it's angry at me because I don't have my uploads yet. Oh, here they go. Okay, well, that FASTQ processes. Let's scroll down here. Or no, we can get started. Okay, so first thing it wants is the FASTQ file. So it's automatically selected that from my history because it knows what file extension to look for. So we're good to go there. We already said this was a 500 base pair amplicon, right? But we want to accept reads that are up to 2% longer than that. So I'm going to put in 510 as the max read length. Okay. And then the minimum read length is, that same length minus the largest deletion minus 10%. So that gets us from 500 down to 350 down to 315 is what we're going to put in there. Feel free to modify those numbers however you like. And our minimum quality score is going to be 20. We're going to be pretty lax on quality here for the sake of having more reads to analyze. Then it wants the wild type DNA sequence. It knows to look for a FASTA. So that's the only one that's in my history right now. So we're good to go there. And then we need the text input for where we want to cut these reads off. Let's go back to the Word document here. And on the 5' end, we're going to put in that sequence. On the 3' end, we'll put in this sequence. Copy and paste. All right, and that's all we need to do there. We click Run Workflow. And we're off to the races. Again, depending on the traffic here on the Galaxy site, this may take a few minutes, this may take a few hours. But it is nice that you can just set this up and walk away from it and then come back later and it should be done. Whatever you see in the workflow that's green, those are things that are complete. Red are things that's in progress. So you can see there it just translated my wild type DNA sequence to an amino acid sequence. And then it started working on the next task here, which is filtering those reads. All right. So I'm going to pause the video here and we'll resume whenever it is complete. All right, and we're back. I know we skipped in time there, but that wasn't bad. That was only about 3 minutes that it took all of this to run. So you can see on the right side here, my history is now populated with several different files. These are the intermediate outputs of all those different tools. So the FASTQs that are modified, the BAM there from Minimap, and then all the FASTA files that follow from there as we convert those into different things. And then finally here, the BLAST-P output, where we've taken all of our reads, translated them into amino acid sequences, and now we're aligning them to the expected or the wild type amino acid sequence. So we can left click on that. We can do a few things here actually. We can view those results by clicking that eye icon. All right. And here we go. So it looks like something did work. We got a lot of different outputs there. Now, if you look at column 1 though, you can see that we did lose quite a bit of reads. So you see, for example, here it starts at read 117. So about 100 reads from this file were truncated there at the beginning. We're also missing read 119, 120. But this is just a fact of life with nanopore sequencing. Since we did apply those filters, we expect to lose quite a bit of reads here. But you see, it's not so dire once we get past there, okay? And actually, if you look here, it says we still retained about 3,000 reads out of what were probably 5,000 to begin with, okay? So that is a lot, definitely. We would have preferred to keep more of those, but you got to get rid of the truncated reads, the ones that are too long, etc. Okay, so the other thing we can do here is download that file. This is a tabular file, okay? And yeah, let's just save that here in my sequencing results folder. And then we can go to that folder. And let's open up Excel. Lots of ways you can do this. You can import that data here, or what I like to do for its simplicity is just take that file and left click and drag it into an Excel window, and that will import it as well. All right, so here's your output blast data. Now here, there's no headings, but I've created an Excel file that we can take all this raw data here, copy it from here into that Excel file, and that will simplify our analysis. So I'm just going to click Control A, highlight everything, Control C, and now let's go over to this other Excel file. Here we go. And we're going to paste all that data into here. Now, in this Excel file, first things first, it has all the headings for these files, or for these columns. So now we know what all these numbers mean. All right, so there's the read number, the subject that it was aligned to. In this case, it's going to be the same for everybody. The percent identical matches, 100% there for the first one, so on and so forth. Okay, but you should be careful about some of these headings. So, for example, that percent identical matches here. What that number represents is if we go over to the right, yeah, here we go. You've got two important columns here. This is the aligned part of the subject sequence. Over here is the aligned part of the query sequence, okay? So you can see here they're not all the same length, even though they all should have been the same length as far as DNA sequence goes. And we should have gotten 81 amino acids out of every one of those reads. All that BLAST is reporting here is the length of the amino acid sequence that aligned to the wild type subject, okay? So it might say that there's 17 amino acids, or actually here, let's do this correctly. If we look over here, here's our length of subject. Okay, it's 81 amino acids long. And then over here, The length of the alignment is 81. There's no mismatches, no gaps. This looks like a wild type read, actually. Okay. Yeah, it's got 81 identical matches. So that first read we know is absolutely a wild type DNA sequence that is giving us the wild type amino acid sequence. But we come down here and let's see, is there an example of where there's 100% score that's shorter than 81 amino acids? Anyways, I won't waste your time here. They do exist. I just want to say that this score here is just the identity in the aligned sequence, not the total sequence. So that's something that we have to calculate for ourselves, okay? So what I really want to know here is every one of these sequences should be around 81 amino acids because of the way we trimmed them, because of the way we cut them off, okay? I want to know out of that total possible 81 amino acids, how many of these residues match the wild type sequence and how many don't. Okay, that's something that BLAST does not give us. So we're going to have to calculate that for ourselves. And so that's what this analysis tab is over here. The analysis tab takes the important columns from the first tab here. So the length of the alignment, for example, the number of mismatches in that alignment, the number of gaps in that alignment. So let me just show you what that would look like here. A gap is obviously one of these hyphens. So you can see we absolutely got some of those deletions here, where we're missing the intervening sequence, but we have what flanks that on the 5' and 3', or the N and C terminal sides, okay? So those are your gaps. As you scroll down here, you're also going to see just occasionally there's these mismatches. Oops, there's a start or a stop codon that's in place instead of that glutamate. Let's go down a little bit farther. There we go. What was that? Here's a valine instead of the expected alanine. So that would register as a mismatch. This read specifically would have that mismatch and some gaps. So we come over here and confirm that, and yep, we see those here. Okay. So coming back to the analysis tab, the length of alignment is going to be the total length of everything that BLAST can match to that target sequence. It's going to include any mismatches and any gaps. So just because it says 81 here, for example, that doesn't mean that that alignment is perfectly matching our subject. Now, in this case, there's actually one mismatch in there, okay, but zero gaps, or zero, gaps where there should be a residue, but there wasn't one detected in that sequence. Up here, the length of alignment was 63, but there was one mismatch in there, and there are 15 residues that are missing. Again, come back here, that's this second read. Yeah. Or maybe it's this one. And you can see several bases missing. Okay. So what we're going to do here is take that length of alignment, and we're going to subtract any mismatches and gaps. That's going to give us the true length of or it's going to give us the true number of how many of these residues in that amino acid sequence actually align perfectly to the wild type. And we're going to divide that here by 81. 81, here, let me change that actually. I want to make that a user input. 81, oops, can't forget my $sign. Okay, now we're good again. So length of alignment minus mismatches minus gaps, divided by the maximum length of alignment there, which would be 81. So if this is a wild type read, like this one right here, you can see the length of that alignment is 81. It has no mismatches and gaps. It's going to score a perfect 100% here. That's going to be scored as 1. Okay. This read here has a few problems. First of all, it's only 29 amino acids long. So only 29 residues aligned. 6 of those were mismatches. So really, it's only 23 residues here, okay, that aligned. That divided by 81 gives us a pretty low score here. So the farther away you are from 1, the worse of a read this is, okay? Now, it should be mentioned here that there's another column, the length, okay? Now, this length here is the length of the read that we tried to align. And I do want to caution you here, one thing we're not controlling against is you see how the total length's only 33? So this is going to show up as a pretty bad mutant, but really, it's only a few bases longer than that alignment. So this could be a truncated read. All right. So we do want to be wary of any of those types of situations. Okay. And we might actually want to eliminate reads that are below a certain threshold. Okay. Because they're too short to even detect some of these Cas9 mutant sites. Okay. So anyways, with that in mind, the next thing we're going to do is, first of all, it's an Excel sheet. This is designed to go up to 5,000 bases. We don't know how many are, so up to 5,000 reads or 5,000 rows. We don't know how many are in here. I'm just going to highlight all these cells that have calculations, double left click there. There we go. And now we've updated these cells for this specific data set. If you scroll down here, I've built this out for 5,000. There's going to be a lot of reads or a lot of rows that don't report anything. That's fine. We'll account for that. But now everything's good to go. The plot here doesn't look that great because we still need to sort the data. So I'm going to take those cells there, or actually, best way to do this, left click in column A, drag over to column F, hit data, sort, and we're going to sort by what's in column F, score. And I want to sort from largest to smallest. Click OK. And this is what we have. Okay. So now we can take these scores and analyze them. That's what these cells are for. First off, this wasn't a great read. Looks like we've only got 655 reads that are in here. And why is that the case? I thought it was 3,000. Wait a second. Oh, I know what I did. Control Z. Here we go. Let's get back to here. I highlighted these cells and double clicked, but that didn't go all the way down, did it? Let's scroll down here. No. Oh, yeah, here we go. See, it's starting at 2 because I was playing with this earlier. Now we've got it. Okay. That should be the length of the alignment. It should be column E, column Q, column W. Okay, now we should be okay. We should get many more reads there. Yeah, there we go. Okay. So what happened there was I just didn't have this formula copying all the way down or accommodating all of this data. It was only 600 reads, not all the 3,000. So now we can come in here, highlight columns A through F, sort by score, largest to smallest. And now we've got a much better looking experiment. The one before, oh, that was scary. Okay, so like I was saying, now that we've got these scores, let's do a few things. First off, we use the COUNTIF formula in Excel to tell us how many of these scores are above 0, okay? And we want to get rid of any that are above 1. Just in case there were any reads that were longer than the 243 bases, that really shouldn't be possible, but just in case, I have that in here as a safeguard. So we're starting at a number of 5,000, okay? That's what the COUNTIF greater than 0, or sorry. We have 5,000 cells here. This is the better way to say it. We have 5,000 cells, but a lot of them are zeros, right? Because we don't have data for those. So what I'm asking Excel to do is count all of the rows that are greater than 0. That should be the ones that actually correspond to reads here. If any of them are greater than 1, that would be an error, and we need to get rid of that, hence the subtraction there, okay? So it's telling us we have, out of a possible 5,000 reads here, we have 3186, and that corresponds well with our graph too. You can see it drops off to 0 there at around 3186. We can also use that COUNTIF function to ask Excel how many of the scores in this column are equal to 1. That would mean that's a read that is exactly 81 amino acids long, so the entire length, with no mismatches and no gaps. So here we see we have about 923 perfect reads, okay? And the plot agrees with that there. You can see just before 1,000, we go from 1 to something slightly less than 1, all right? So if we are being really, really stringent here, out of 3,000 reads, about 1,000 of them are perfect wild type, okay? But remember, nanopore sequencing sometimes makes mistakes. So here is where things get a little fuzzy, okay? And we need to know that that might not be the actual number of wild type reads. There might be some reads that are actually wild type, it's just they have one mistake. So the sequencer made a mistake on one base that changed one of the codons to correspond to a different amino acid. It's absolutely possible that happened here. So it's worth noting that about 200 of these reads have a single error, and they're ambiguous at this point. They could either be wild type or a mutant read with a mutation at one or more of those Cas9 cut sites, okay? So we can ask Excel to calculate the number of reads with just one error. That's what the formula here does. How many reads have 2 errors, 3, 4, 5, 6, or greater than 5 errors? So you can see things are a little fuzzy here to begin with, but then there's very few events or very few reads that have 4, 5, or 6, okay? So what do we do with this? How do we estimate a KO efficiency with such ambiguity? Well, what I would advocate for here is one of two things. One, if somebody more well-versed than me in Galaxy or writing code could go through and look at each of these reads, there's probably a clever way to look at those mismatches, okay? So for example, this one right here, where we have a stop codon instead of a glutamate, and ask if that mismatch is either random or if it might correspond to a Cas9 mutation. What I mean by that is you can see here that it occurs at this glutamate. While if we go back to our gene sequence and look at our Cas9 targeting data, we don't expect to see a mutation that early. The first mutation should happen here at this HAQM motif. So like you can see there's several reads where that's the beginning of a deletion because Cas9 cut there and then somewhere downstream. So since this mutation exists so far away from that Cas9 cut site, it's probably not a mutant read. That's probably just a mistake on the part of the nanopore sequencing or the PCR. Okay? So I think there is a better way to do this um, by taking that into account. But that would take a lot of work that I haven't built into this sheet yet. So the other alternative that I would advocate for here is just calculate the knockout efficiency for every one of these possibilities, okay? So what I'm giving you here is a best and worst case scenario. So I can say here, if I calculate my knockout efficiency to be all of these reads that have at least one error, okay? It would be 71% of the reads have at least one error. So you could say that's your knockout efficiency. If you allow for one error though, and you calculate your knockout efficiency by only adding up the reads that have two or more errors, so it's the sum of these cells divided by the total reads, then you get something that's closer to 65%. And so on and so forth. If your threshold is 5 errors, you would sum up these reads divided by 3186 to get that. So overall, I think we still get a pretty clear picture of what's happening here. The knockout efficiency is somewhere between 60 and 71%. This is a case where we could probably do a better job with our Cas9 knockout, right? So we have 900 reads here that were perfectly identical to the expected sequence. So if I wanted to make sure I had a more homogeneous population in which all of those cells were missing the correct function or correct and functional version of this protein, I'd probably go back and tweak something. So add more Cas9, add more guide RNA, maybe change the electroporation settings if that's how you're delivering this, so on and so forth, right? So this is not giving me something that's very quantifiable due to this, uncertainty here with the nanopore sequencing, but it is giving me a rough view of if I need to go back and change my KO settings, okay? And in this case, it looks like we could do better, all right? So go back, change something, do this again, and see if all of these numbers go up, right? That would be a good sign that you're optimizing your protocol. Okay, so that's it. Hopefully that's enough to get you started analyzing this nanopore sequencing data and getting some idea of how many of those genes you've knocked out. Have a good day.